carbohydrates, carbon, hydrogen, um, eukaryotes, prokaryotes, viroids, red blood cells, white blood cells. How all these different size of cells, how they are compactly, very nicely maintained in our body, and they are sized from millimeter to nanometer to picometer to micrometer. So all these different sizes, and they are being very nicely done in our body. So this is small presentation being showing here. So in our WordPad, when we are writing, it's mostly times regular 12 point, yeah. And the coffee bean size is around 12 by 8 millimeter. And the grain of your rice, it's around 8 by 2.5 millimeter. If we go more deep, now we're gonna zoom from millimeter to micrometer, slowly and slowly. Okay. So sesame seed is around now 3 by 2 millimeter. Now grain of salt is around just half a millimeter. Then comes your prokaryotes, amoeba, in the micro range of 500 micrometer, paramecium by 210 by 60 micrometer. Yeah, the human egg around 130 micrometer. Our photoreceptor that are present in our eyes as cells, 2.5 to by 100 micrometer only. And skin cells around 30 micrometer, and sperms around 60 by 5 micrometer, and RBCs are around 8 micrometer, chromosomes around 7 micrometer, and Baker yeast by 3 by 4 micrometer. Then mitochondria is around 4 by 0.8 micrometer, E. coli bacterium. So these are all bacteria, protozoans, prokaryotes. They are all into the micrometers size. Now afterwards comes your viruses, which are of nanometer size. That COVID that we are seeing at the moment, of the size of 220 nanometer, influenza virus of 130 nanometer, HIV around 130, phage 70 by 200, like so on. Hepatitis 45 only. Then comes various RNA and ribosomes, your tRNA antibodies. So they are also into the nanometers of like 30 nanometer of ribosome, tRNA around 7 nanometer, antibodies of 12 nanometers. Heme, your hemoglobin, just mere 6.5 nanometer. So you can see all things being comparable here. Then we go even further. Now things will be into the picometers. Your nucleotide, your nucleosides, your amino acids, they come into the picometer size now. That is 10 raised to the power minus 12 meter. So water molecule, glucose, they're around 1300 to 760 picometers. And at the end, carbon atom, from where the life starts, it's around 340 picometers. So we can say, as a basics of our life that has been started, that we are originated, is basically from carbons, oxygen, nitrogen, and all these sizes you can see so tiny, 340 picometers. And gradually they become together, they go for into the nanometer size, then micrometer size, then yeah, millimeter size. So that's the, yeah, cell and size, cell size and scale, how they make an impact on our body. So I will keep sharing these into two groups. So far so good. Now the next topic I want to initiate, in continuity with the previous thing that we have discussed, that is cell size and its scale, to discuss more about cell as an analogy to the factory. That how in a factory all the people, all the assembly line people, transporters, security guards, CEO, secretary, everyone are working together in order to have production. whether it's a Coca-Cola bottles or Nike shoes or I don't know Gucci purse, anything. So the production is all effort of everyone, each individual from each compartment. And if any compartment is not working in synchronization way with the other, so things will not work as it should do. The same way in the cell, if your mitochondria, Golgi bodies, ER, nucleus, cell membrane, microtubules, ribosomes, any of them, they are not synchronized with each other. So the protein production will be not there, and hence there will be some disease start to accumulate because of the degradation of protein. So everything needs to be work synchronized in order to have nice production. So how can cell be a factory like? So in a factory, we have many different tasks being completed in the same building, like building sports, as we have discussed just now, workers building goods, maintenance crew, clean up messes, shipping departments, good ready to leave the factory, and so on. So many things has to be taken care in the factory. The same way, the cell has to perform different works in this, different compartments in order to fulfill their functions. The cell wall is like a security gate. So first of all, cell wall. So a security gate protects the factory and helps keeps it safe. Yeah. A cell wall does the same thing for a plant cell. So like this, you can open it up and close it up. So whatever the things need to be come in or go out, like sodium, potassium pump, calcium pump. So all these things have to be carried out, which via aquaporins, your water molecules are moving in and moving out. So all these pores and channels that are helping in to let things in and out, acting help as a cell wall. Then comes cell membrane. This is after that is the front doors of a factory. So once we have these big doors, then comes your front doors, which regulate and enters the building of our staff, who should come and who should not. So it's the same thing for plants and animals. In both it is present, but the cell wall is mainly in your plant's presence, not in the animal cells. So like this, someone is knocking the door to enter the factory. Nucleus is the CEO, the major boss, big boss, who controls the idea behind it, how a cell should work, which protein it should produce. So it's a big boss, the CEO, controls all the activity inside the cells. And nucleus does the same thing for plant and animal cells. Yeah. So like this is the big boss, the CEO, recording being there. And cytoplasm is the factory floor. The floor of the factory has all the workers, machines, and equipments on it. Yeah? And the cytoplasm holds all the organelles in the plants and animal cells. So like this. Then endoplasmic reticulum, that is the ER. So an assembly line where the workers do their work. Items move along the conveyor belt to different parts of the plant. Yeah. So ER moves items to different part of the cells in the same way. So like it helps in the moving your things within the cells. So the workers are working and your assembly line, your products are moving, like the CD disc or some bottles or some containers. So in this way, ER is working out. Then ribosomes, which are workers on the assembly line. So they are assembly responsible for building the goods in a factory. Um, they help to assemble your proteins, your small ribosomes, big ribosomes, 70S unit, 80S subunits. So they are helping to produce your proteins together with the help of tRNA. We will discuss this in detail about today in the basics of molecular biology with animated video and also with the presentation. So in this way, it's helping us to build your things. Then vacuoles, they are the break rooms in which you have to storage for food items and places to get rid of trash. Vacuoles hold waste and stores water in plant and animal cells. Which cell has a large, has one vacuole? So like we can see here, everyone is coming out, your food and everything is being stored in this vacuole. Mostly they are present in both plant and animal cells. Then lysosomes, they are the maintenance janitor crews who take care of the things of repairing things. If there anything break up or anything need to clean up, so your lysosomes comes and play a role there. Yeah, around the factory, how things need to be done. So lysosomes do the same things in the cells. So they are the one who take care of all the electricity, wooden furniture, everything at one go. Then mitochondria and chloroplasts, so they are the energy source of the cells. ATP production is being done there. So power station which supplies energy to the factory. So your mitochondria and chloroplast in the animal cells and plant cells, they do the same for the cell. So this is the example of the factory looks like. You have a ribosomes. Yeah. So first of all, there's a gate cell membrane. Then comes your so after the cell wall, there's a cell membrane. Then comes your endoplasmic reticulum nucleus, from where your main CEO is working, and then the Golgi apparatus is there in order to have the waste products coming out. Then productions of protein is being there with the help of ribosomes, and mitochondria is helping to produce energy outside there within the cell. So in this way, everyone is working in synchronizations and um, making things done good. So we can say that in this small example, that we just want to refresh our memory about the cells, so that we get to know about the where is the transcription is happening, how DNA is moving from nucleus to cytoplasm, which is playing which role. So in that way, this will might refresh our memories about the cells. So this is not for you. Yeah, that's it. I should remove this. Yeah, sorry, this is for you actually. You have to just make one example in the assignment with cell as a factory with an example from any uh, message. No, from any ketchup factory, shoes factory, how the bottle of ketchup or the pairs of shoes are being produced in the factory. So you can take an example and draw a one page figure, one page assignment out of it. Yeah. So as I have discussed last time, so I will share these documents with you as soon as we are done with them. So you can have your study material with you. So cool. Okay. Just take a 1 or 2 minutes break here, then we start the next part. The basics of molecular biology. Okay, I use this presentation. This is far better than the previous one. Just to explain you structure of DNA. Forget that. I'm not anymore part of it. Just take a break, then we start the next class. Okay, so we start with DNA first. I hope you all aware of nucleotides, nucleosides, bond between two nucleotides, purines, pyrimidines, bond angles, different types of DNA, different types of RNA, you know. So we might discuss now this today. Just to give you basics of first molecular biology, then we go to the advanced topic then. So as we know, basically the DNA, which is the main, uh, strongest backbone of our cell, from which everything is governed. Like a building, if there is a big building, the basic foundation of that building is the bricks. So one brick joined with another brick joined with another brick, and another, and on top another come, and on top another come, and top another come. And these bricks actually are the nucleotides named with like 80 CGs. A, T, C, G. And correspondingly there will be another thing. So basically ATCG, they are the basic foundation of DNA, in which they are packed into the nucleus quite compact and in a very nice way. So, and these together they explain the central dogma. So we will discuss about also central dogma, which sometimes we tend to forget. We keep studying. This happens to me. Why should I lie? That I keep studying on PhD. Then one of the interview at the government institute, they just asked me what is central dogma, and at that time I was just out of my ways, because you are so much into your own studies that the 11th, 12th, your BTech and all these, you tend to forget all these things. So it's always good to rebrush our memories. It's always good to learn and relearn the back things. So first of all, history. So who was the one? Friedrich Mischer in 1869. He discovered the nucleic acids initially. Then the Griffith experiments he did with this bacteria, if you remember, in order to with the and smooth, rough and smooth coating of your bacteria, in order to find that the transformation is happening. Then that was not enough. Then Rosalind Franklin, he got the first X-ray photograph in 1952. But later on, the big discovery in 1953 by Watson and Crick, they took the Franklin X-ray and they described the DNA molecule. But I would say the key point, the key master was Rosalind Franklin, who have discovered the X-ray crystallograph. So it was something like this. The X-ray, it was coming like this, of the, if it is the image, then the image of the DNA was on the X-ray was coming like a cross, and in between they were seeing these some nucleotides being joined together. So this was very remarkable, very unique for all of us to see, and they depicted all these things, that what it was. But Rosalind Franklin was not able to depict what was it, why the structure was helical. So all these things were depicted by the Watson and Crick. Then there came Chargaff rule, which says your purines and pyrimidines are in equal amount presence. There is no way that they should be in different proportion. So DNA structure, as you can see in your right hand side, very nicely bubbly structure. So sometimes whenever in the exams, when it comes to DNA structure, including me, we used to just, okay, DNA structure is there, we have to make this this way, and then we go like this, yeah, and then we go like this, and this that's it. And then we join with the nucleotides, yes, we join with the hydrogen bonds, yeah, this is our DNA. It's never been, DNA never looks like this. So you can see in this structure that it's being parallel, things are going in parallel and in this slant way. So always whenever it comes time to draw your DNA structures, always tend to go in this ladder manner, like we have in our circular ladders in our houses. So this way, yeah. So like this, like this. So keep in mind while drawing this structure. Yeah, so DNA is a double helix structure with two chains having sugar phosphate bonds. So we are having it here sugar and then phosphate bonds outside and the nitrogenous base pair inside. Inside is the nitrogenous base pair. So these are all your nitrogenous base pairs, and outside is the sugar and phosphate backbone. And the molecule of DNA is made up of millions of tiny subunits called nucleotides. So as we have discussed in previous in the first page, that it's many, this ATC as a brick of a building, they are the main reason for the DNA. So they makes the basics, millions of these tiny subunits make the foundation of your DNA. So in prokaryotes, they are called as nucleoids and a plasmid, and in the eukaryotes, they are called the chromatin of the nucleus. So prokaryotes and eukaryotes have different formation of it. We will discuss them in later. And adenine always pairs with thymine, and guanine always pair with the cytosine. Yeah. So A will pair with a double bond, thymine. I know most of the 80 to 90% students knows about this. You are the cream of India here in our session, but it's my task before I go to the advanced levels. The basics need to be clear first. So A and T will make the double bonds. G and C will might make that might not. Sorry, tongue of slip. Will make definitely triple bonds, and these are hydrogen bonds. Okay. So DNA structure, what is the basic structures looks like? It has nucleosides and nucleotides. If there is a ribose sugar, which is a deoxyribose sugar, which doesn't have on the second carbon your oxygen, and there are four different base pairs, nucleotide base nitrogenous base pairs, A, T, C, G. Yeah. And here we have a phosphate group. So this is sugar, deoxyribose sugar, then ATCG. So together is the main component of it. And difference between your ribose, the RNA, and your DNA basically is your second carbon, prime carbon. So some students were asking me in my previous lectures that, sir, what is this prime? Why we call this prime? This is the nomenclature, like the carbon at the first in the chemistry. This is how we name it. So this one is the carbon 1 prime. This is second prime, this is third prime, and this is fourth prime, and this is fifth prime. Yeah. So at the second prime end, in the ribose sugar, our hydroxyl group is present. In the rest, everywhere hydroxyl group is present. OH is present here, OH is present here, OH is present here, OH is present here. In the deoxyribose, in the second prime, at the second carbon atom, your hydroxyl group is not present, OH is not present, and that's why it's called deoxy. So that's why DNA, deoxyribose nucleic acid, and this is ribose nucleic acid. That's the main difference. Then DNA structures. So the bases, basically, as we have discussed in the Chargaff rule, your purines are equal to the pyrimidines. So adenine is making bonds with thymine, and guanine is making bond with the cytosines. So equal amount of purines will make equal amount of pyrimidines bonds. So the nitrogenous base pairs, they are divided into two parts, that is your pyrimidines and purines. Good. And the pyrimidines, they are made of one single member ring. So always remember, pyrimidines, they are the single 6-member ring, whereas your purines, yeah, they are both fused 5 and 6-member ring. So there's a 5-member ring here, and this is 6-member ring here, and they are fused together. And this is 6. And in pyrimidines, you have TC, yeah, thymine and cytosine. And in purines, you have AG. You can remember something like short form puri, like your one wife is asking a husband, AG, have some puri or something. So you can correlate with that. Adenine plus guanine. And pyrimidines, they are the TC, thymine and cytosine. Remember them, sometimes they come that we tend to forget in our objective types, that how much purines should be present if this much of pyrimidines are present. So you need to see, count that how many thymines are present, how many cytosine are present. As much as thymine and cytosine present, it will be the equal amount of your adenine guanine is present. So this is very good for your MCQs types of questions. Then comes rings are not only made up of carbons, specific formula structures are not required for. Um, this called nomenclature, yeah. Okay, now this one, the purines and pyrimidines. Now this is how we have discussed since the start of our class about the basics of DNA, how it looks like, and we slowly gradually started that it has three main components: ATCGs, nitrogenous base pair, sugar and phosphate. And we have discussed two types of sugars are there, and then we have discussed purines and pyrimidines. Now it comes to make like a puzzle. It was to join them together to solve the puzzle. So what happened that you have these purines, adenine, guanine, 5 and 6 membered rings, and they are being joined to the sugar here, yeah. And these sugar between each other, they are joined by the phosphate group. And the bond, remember always the bond between sugar and phosphate, it's called phosphodiester bond. This is being asked many times. We tend to students tend to forget this also, because things are very easy. It's just we make them complicated. These are phospho, sorry, I don't have a writing pad, soon I will get it. Phosphodiester bond. Just using my mouse to write. Between sugar and phosphate, and the bond between your sugar and nitrogenous base pair, this bond is known as, anyone? Yes, we will discuss, bache, Shalini, we will discuss. And the bond between your sugar and phosphate group, it's known as, anyone? Yes, very nice, very nice. Anyone else? It's glycosidic bond. So in this way, sorry, this is bond. Our backbone of our, and on other side you will have another nitrogenous base pairs which will be attached to the another by the hydrogen bonds, and we will discuss them here soon. So this way our DNA is made. This is deoxyribose sugar, our phosphate group is there, our nitrogenous base pair is there. And in case of this is in the DNA. In the case of RNA, what will happen, instead of thymine in your pyrimidines, you will have uracil. You have a uracil. In RNA structure. And in RNA, you will have, instead of deoxyribose sugar, you will have mainly your ribose sugar. Okay, now we have discussed the basic foundation of our DNA. Now comes the next part of nucleosides and nucleotides. So what is the main difference between nucleosides and nucleotide is that in the nucleosides, you have mainly the nitrogenous base pair and the deoxyribose sugar. And in the case of nucleotides, you have three things, that is your deoxyribose plus base pair plus phosphoric group, the phosphate group. So together they make nucleotides, and without phosphate it's make a nucleoside. So that's the main difference. So we have given these this nice table: adenine, guanine, cytosine and thymine. So If the base is present, that is your adenine, and you have a deoxyribo sugar, so the nucleoside pair will be called as deoxyadenosine. That is deoxyribonucleosides. But if your phosphoric group is present within this deoxyribonucleic side, it will become a deoxyribonucleotide. So we will put T at the end, and here we have S at the end. So deoxyadenosine, and this name will be deoxyadenosine plus monophosphate, because phosphate group is added. So within this we have added a phosphate group, and the short form of this will be DAMP. And for the guanine, it will be a deoxyguanosine, yeah. And for cytosine, it will be deoxycytidine, and for thymine, it will be deoxythymidine. And the same goes like for the nucleotides and their abbreviations. Cool. So that was the difference between your nucleosides and nucleotides. Now comes the next part. We're gonna discuss about, we have discussed this already, that the nucleotide structures includes your phosphate group. So, and these together we have discussed in our previous slides. So one nucleotide together will join with another with the help of your phosphodiester linkage. With the help of phosphodiester linkage. And DNA double helix and hydrogen bonds. So two strands of nucleotides, as we were discussing, they are joined together by your hydrogen bonds. And adenine is always paired with thymine, and cytosine pairs with the guanine. And hydrogen bonds always occurs between pyrimidines and purines. So one pyrimidine has to be there, on the other side one purine has to be there. So adenine and thymine will make double bond, and cytosine guanine will have a triple bond. So this diagram shows quite apt summary of what we have discussed so far. Initially, in order to understand the DNA strand, it runs anti-parallel towards each other. What do you mean by anti-parallel? Anti-parallel means the starting of one strand, if it is starting from 5' end, it's ending at the 3' end. Yeah. On another side of it will be starts at 5' end here and will end at 3' end. And they both will be opposite sides of each other. So here we can see a phosphate group is there, yeah. And if we see here the glucose, the sugar group present, so it will have 1, 2, 3, 4 and 5. Here is our 5th carbon is present. So that's why it's the 5' end. So at the 5' end, it will always have a phosphate group attached to it. Yeah. And then at the end, it's keep going running with the deoxyribose backbone. One sugar group is attached to the other phosphate group, and goes. And here also 1' end, 2' end, and 3' end. So at 3' end, your hydroxyl group is there. So here your this is getting over. On other side, the same way, 5' end is here, and here is in the 3' end. So that's why this is an anti-parallel. Cool. So this is an anti-parallel backbone. And second thing is how adenine and thymine why they are making disulfide bonds, because there is no possibility of making triple bond there. It's only these two nitrogenous group, these nitrogen group making with this hydrogen, and this nitrogenous group with making with the hydrogen, oxygen and hydrogen. So only 2 bonds are possible here. Whereas in the case of guanine and cytosine, triple bond is possible. NH2O, NHHH and H2N and O. So there possibility of 3 bonds, because their questions might come that why not adenine make triple bond? Why not adenine make bond with cytosine with C? Because it's not possible, because it is a tendency to make triple bonds, and adenine has only tendency to make double bonds. So in the nature, what is in nature is present, it will be like that. We cannot change to it. Yes, there's one thing that mutation could happen. So thymine, if there is thymine here and we put also thymine here, so they make dimers here, they make double bonding here together. So this is possible. It's just because of the UV light. In the DNA repair system, you study about that. But in this case, everything in the nature, things are being present like this. So the strand runs opposite to each other. At the 5' end, we will always have this phosphate group attached to it. Yeah? Never ever forget that. So all good so far, students. Okay. Oh, my Taiji is there. Thank you, Taiji. Just giving me thumbs up. Then Watson and Crick model here. Here we can see that in the Watson and Crick model, so whatever they have discovered so far, so now they want to bring it into the reality, what it looks like, because anything has to be developed, anything has to be come into the reality. It doesn't come in a day. We have to first of all cut out its blocks. What is so, like, they have discovered these, first of all, glucose sugar groups, then they have discovered these phosphate groups, then they have discovered this nitrogenous group, and then they have discovered the bondage between them, and then they have discovered the helical structure, and then they have discovered its angles, how the angles are being done. So each steps that we have discussed today within this chapter of DNA, it doesn't come in a single day. So any research that you're doing, any things that you will be doing, pursuing in your life, it will not come in a single day. So you need to, first of all, in while researching or anything that you're gonna do, break it into simple manner. The more you make it simple manner, the more it will be easy to understand, and more easy it will be explain it to the audience also. So that's the beauty of science is. That's how scientists think when expressing their experiments, that it should be explained in a way that the audience could understand in easy manner. That should be the focus. So in the Watson and Crick model, as we have discussed so far, so adjacent deoxyribonucleotides, they are joined in chain by the phosphodiester bond with the 5' end of one carbon of one of the mononucleotide with the 3' carbon of the deoxy of the next mononucleotide. So if we have 1, 2, 3 here, so this is 5. We have discussed this point also. It's just I brought these points in each slide, so that this is 5' and this is 3'. So 5' is attaching to the 3' end of your carbon. Further, in the same Watson and Crick model, so two chains wrapped helically around each other with sugar phosphate outside and base inside. We have discussed in the first chapter that outside sorry outside are your sugar and phosphate group, yeah, they are being attached with each other, and inside is your nitrogenous group present. Hydrogen bonds are being made between each other, yeah, hydrogen double or triple as per there. Two chains are complementary each other, so one will go 5' to 3' end, yeah, that is their anti-parallel. Other go up 5' to 3' end. And calling of DNA is right-handed. If I could make my movie up, if you have a right thumb, if you just make your thumb up, and you will, when you folding uh, your fingers, your four fingers, they will move into the right-handed way. They will move into the anti-clockwise direction. So that means the right-handed calling is done in the right-handed way. And one complete turns have around 34 angstrom. So they are major groove and minor groove. We will discuss them in later. So that including your one major groove and one minor groove, the whole distance, the nitrogenous base pairs includes, it's the distance is around 34 angstrom. The whole distance including your the longest length of your DNA, including your major groove and minor groove. That distance is known as to be 34 angstrom. And during this base pairs, this one turn it includes 10 nucleotides. So your 10 nucleotides are present between during this one turn. And this helix, as I just discussed, major groove and minor groove. So this is the very nicely shown two models of the DNA structure. That is a ribbon model structure, and then there is a space filling model. So the diameter of a typical DNA, it usually go for around 2 nanometer. Yeah, the diameter is around 2 nanometer, and the major groove, as have we discussed, which includes your it's taking a big turn here, you can see, and then going back. So it's quite deep. Yeah, it's quite deep and thick. So this includes your double stranded, and the size is of around 2.2 nanometer. Whereas the minor groove, it includes a size of around 1.2 nanometer, which is quite thin and not so deep, your minor groove. And your total major groove and minor groove, that together make one length, and this is the size of around 3.4 nanoangstrom. Because remember these things as a major groove and minor groove, because this will help you to differentiate between your DNA A, DNA B, and DNA Z, because that's the main difference between the three structures. Actually, that's what the three structures of your DNA are differentiated between each other. Yeah, that we will do in the next class, not today, because this will be too much for all of you. So we just yeah. So here we can see A, T, C, Gs. They are showing double bond, triple bond, purines and pyrimidines, nucleotide base pairs, and on the outside the sugar phosphate backbone is there, and this is a central axis there. Yeah. Um, yeah, that's it from here. So the features of this DNA double helix structures. So the two polynucleotide chains, they coil to form right-handed helix, as we discussed. They run from 5 prime to 3 prime end and other into opposite directions. So nitrogen base pairs are opposite chains pair to one another to form hydrogen bonds. Base pairs stacks on each other located on the inside the helix, and one complete turn is around 3.4 nanometer, including your 10 bases. And each turn includes your one major and one minor groove. Yeah. So whatever I have discussed just now in this thing, it has been summarized in these points. I hope by today, your features of Watson and Crick model might be clear for all of you. Some of you might be aware of it. I know, but it's good to have a strong starting point, and then we go to the more detailed parts later. Cool. Any questions? The last 5 minutes will be the 4 questions answers around now. If you have any questions, feel free to ask me from today's lecture, or anything else beyond studies, or anything, um, review article, assignments, or anything you want to ask. Yeah, Kaushika Lohia. When sugar bonds with phosphate group in the same nucleotide is called phosphodiester bond, where the sugar bonding with phosphate group of adjacent nucleotide bond is phosphodiester bond. I didn't get you. Yes, you are correct actually. Okay, so student, we will continue with this DNA part tomorrow in more details, and then we will have some live, some more animation classes tomorrow regarding the same. So I hope we had a good, you have good knowledge about this today in the class. So that's it for today. If you have no further questions, is there any ratio of ATP molecules with respect to the size of bacteria? Hmm. ATP molecules with respect to, I mean, yes, of course, you will have more mitochondrias if it is present there. The more energy could be stored there, and the more ATP could be produced. So, of course, um, this is possible. Such assignments you can submit later. No worries. Once we are done with the basics parts, then we can do it. Let me take some questions. Thank you, Sangam Desai. Phosphoester and phosphodiester. Shivam Keshirvani. So this is actually the chemistry part. I need to draw this actually. Phosphoester linkage is the like single bond happen between your phosphate group and your sugar group. In the phosphodiester is means that your phosphate group in the center and you have two sorry um, yeah, yeah. You have two sugar bonds there. So this is a phosphodiester linkage. Yeah. And if this bond is only made with the one sugar here and one phosphate group, then this will be a phosphoester bond. But here we have two oxygen groups, and these together they are making bond with the sugar. That's why it's diester. It's become a from ester to become a diester. So that's the major difference between your phosphodiester linkage. Yeah, I can show you this. Yeah. So you have your sugar, CH2, yeah, the regular stuff here. Yeah. Um, Here we just put O, P, O, H, then O, H, double bond O. Here we have no sugar bonds upstairs, so it remains like that. Now, so this is your phosphoester linkage. I need to buy. This is phosphoester linkage. But once you have one sugar, So in this CH2 O P double bond O, just draw in your copy if you can, OH, and here another sugar group will be there. Your base pair could be joined here, nitrogenous base pair, here also. So again CH2, and it will make another bond on top. So this will be at the bottom one is your phosphodiester. Sorry if I'm just using my mouse here, so these things are not good as they should be, but soon I will get my pad, then things will be better. But the major thing I just want to explain that the ester, because the question has been asked, it's very important question when you're discussing about DNA, that why, what is the difference between phosphoester and phosphodiester. So major difference that when the phosphate group is making two bonds with the sugar group here, that's become a phosphodiester, otherwise it's a single bond. Cool. Shivani Bohidar, can we take help of anyone? Yeah, sure, you can take anyone's helps. You're most welcome to do so. Sakshi, this is somehow into the microbiology. Maybe you can just ping me in the question. I will try to answer this. Okay, so that's it. If you have any further questions, you can ping me up. I will be happy to answer them. So I will share this slide to you also, so that you can fresh it up a little bit for tomorrow. So structure of DNA. Maybe I just remove the logos from there, then I will share it. Okay, that's it for today. Thank you very much. Take care. Good night. Bye bye.